Hello, good afternoon, everyone. So now welcome to the manufacturing section. I know over the 1.5 days in the past, we all heard the very impressive scientific talks and also the positive impact of the alpha-beta depleted HSC transplantation for the patient. So in this section, our focus will shift to the manufacturing, something behind the scene, and how we make the cells for the patient. By the way, I'm Gavin Wang from CGM and the Laboratory for Cell and Gene Medicine here in Stanford. So we are a translational research lab and also a GMP facility in Stanford established about 7 years ago. And we are here to support Stanford faculties as well as the industry partners for the CMC need in the cell therapy field, including from the process development to the manufacturing and also the quality control, quality assurance. As Rosa mentioned, so we are the facility to manufacture the TLO product, the Table product, and also the FOXP3, the Treg product. And here we also have a very close partner located inside of the hospital called the Cellular Therapy Facility, also as known as CTF. CTF manages the cell product from the beginning of the apheresis collection all the way to the final step to prepare the product for the patient infusion. And the CTF also have the capacity to manufacture what's so-called the minimal manipulated cell product, and TAB is one of them. So with two groups together and also with some help from another GMP facility in the Bay Area in UCSF, I think in the past 3.5 years, we have manufactured over 100 product for the patient treated here in the hospital. And today, for this session, we will have 3 speakers. So we have our manufacturing lead from both groups, Nick from CTF and also James from CGM, and they will talk about the initiation to set up the manufacturing process and also our ongoing effort to complete transfer the process into a fully automated manufacturing workflow. And additionally, we are honored to have Dr. Papanikolaou, the strategic team lead of the HSC program from Eleni Biotech, and she will provide us some update about the continuous development of the process and the QC analysis. And given the nature of the 3 talks are highly relevant to each other, so for this session, we plan to hold the QA together at the end. So without further ado, Nick from CTF. Thank you.
Hi everybody, my name is Nick Sukle, and like Gavin was saying, I'm the IND manufacturing supervisor at the cell therapy facility. And I'm going to be talking about building a TCR alpha beta manufacturing program. In this presentation, I'm going to go into an overview of our facility, since we are a little bit different than LCGM, and we're going to go over the entire TCR alpha beta process. This is going to be from a high-level overview. I'm not going to get into the details. This is just to kind of show logistically how we do this process and to show new facilities how they could do it themselves. I'm going to get into the technology transfer. So again, if you're a new facility and you're bringing this process in, I'm going to go over some lessons that we learned about our tech transfer and what you can do to make the process better. And then I'm just going to get into some final observations and our future plans. So like Gavin was saying, our facility, we can minimally manipulate cells. We're ISO 8. We have 4 processing bays. And as you can see on this diagram over here, we have 2 bays set up currently next to each other where we can do one alpha beta depletion in each bay. So for us, we do 2 depletions in parallel at the same time because we have the space to do it. We also have our CRF and LNF right next to our processing, so we can freeze right after we finish. And the QC laboratory is not pictured here, but they have BD fax lyrics right now. Not pictured as well, I want to highlight just the rest of the CTF. So there's a whole other side to this laboratory. That's the core processing lab. That's going to be your standard of care auto allos mostly. And then also the cell pharmacy facility, or the cell pharmacy team also works amongst both those labs. So if you have any cell trial coming into Stanford, it's going through cell pharmacy. I'm not going to stay too much on this slide. I included this in case somebody wanted it and I sent out the slide deck, but this is just the process overview from our batch record. Again, I'm not going to get into these details. I'm just going to go over a high-level overview. So on day -2, so day 0 is infusion day. We're going to receive the apheresis approximately 2 o'clock. We're usually getting around 60 to 120 billion cells right now. We're going to pull a cell count and flow cytometry sample, and we're going to dilute the product if we need to, and we're going to store it overnight. About 2 to 3 hours later, we'll get the flow cytometry results, and they'll be reported to me, and I'll fill out this sheet on the right-hand side. With that data, I'll then call usually Dr. Bertena or whoever the physician is, and I'll report the CD34 dose, the TNC, the CD20, and the TCR alpha beta. From those numbers, physician is going to decide how to proceed forward with manufacturing. We can currently load 55 billion cells onto each Clinimax. I'm going to say 99% of the time we're going to do both depletions in parallel, but we do have the capability to do a CD34 selection if we need to. In this discussion, we also might have to increase the number of vials that we add. They could be at or near the Miltenyi guidelines for cell limits. Also included at the bottom of each of these next few slides, I included the equipment that you would need if you're this facility and the personnel. So it would just take 2 operators and the refrigerator and BSC to complete this processing day. The next day, we're going to start around 8 a.m. We're going to split the apheresis, like I said, if we're going to do 2 depletions in parallel. We're going to start a platelet wash on the Lovo. The Lovo is a cell washer made by Scale Ready or Fresenius Kabi. They have 2 names. And the instrument requires volume, WBC concentration, hematocrit, and platelet concentration. Our cell counter gives us those numbers, so if you don't have a cell counter that gives you those results, I would recommend getting one. And also, if you don't have a Lovo, we use the code 2991 for our cell washing for CD34 selections. Miltenyi has a procedure for doing this whole thing on with the centrifuge, and if you have a similar cell washer, you could probably use that as well. We use the Lovo, but it doesn't need to be what you need. So we're going to do our platelet wash. At the completion of the platelet wash, the final product bag is going to, the volume is going to be set. That's per Miltenyi's guidelines for labeling. I'm not going to get into those as well. We're going to add the TCR alpha beta and biotin vial, and we're going to incubate at room temperature. At the same time, we'll have a cell count. That cell count is going to give us the results that we're going to input into the next Lovo procedure. After the 30-minute incubation, we're going to load the cells back onto the Lovo, and we're going to do an antibody wash. We're going to wash out unbound beads and any excess reagent. Again, we're going to repeat this one more time with CD19 beads and antibiotin beads. Wash that out one more time. That's going to take approximately 4 hours. And again, you're going to need, for us, we're doing 2 Lovos in 2 different rooms with 2 different people. So we're doing it all at the same time. Around noon, the final product on that last antibody wash is going to be harvested at approximately 275 mils. The max volume that can be loaded onto the Clinimax is 300, so we're just a little bit below that. We're going to obtain a labeled wash flow cytometry sample at this time, and then we're going to set up our depletion on the Clinimax Plus. We're using depletion 3.1, which is the standard program. After depletion is finished, we're going to dilute each of the target samples 1-to-1, and we're going to store that overnight. We'll also obtain a cell count and flow cytometry sample from each. We also will pool the non-target samples and pull a sample as well. Everything is stored overnight, and again, this is 2 people with 2 different Clinimaxes. The next day, after we receive the results of flow cytometry, my team will come in and they're going to start completing the calculations you see on the right-hand side of the screen. When we get the TCR alpha beta dose and the CD34 dose, again, we're going to contact the physician and we're going to advise them on those doses. From there, they're going to decide how we proceed forward with manufacturing. So it could be any number of different things. It's very hard to write this batch record because there's a lot of different options. So you could infuse all of target 1 and cryopreserve target 2. You could do half of target 1, half of target 2. It could be split any sort of way. So there's other calculation pages where they get into that, how you mix the 2 products. But let's say for this, you're infusing target 1 and cryopreserving target 2. For the target that's going to be infused fresh, day of, we're going to centrifuge the product and we're going to resuspend at approximately 150 milliliters. We're going to take our safety testing, sterility, gram, endo, and once we receive results from that, around approximately 2 to 3 in the afternoon is when we'll get to infusion. During this time, the backup product is going to be cryopreserved. So that's why I said 4 personnel. So you have 2 people doing the infusion, 2 people freezing the backup product. So that's just a basic overview of how the process would flow when you're doing it. But now getting back to, if your facility is bringing this in new and you're starting from scratch, this is how I would recommend doing a tech transfer, bringing this in. So I would start with a process development or training stage, whatever you want to call it. I'd recommend doing at least 3 runs. It depends on your level of comfortability using the Clinimax, using the Lovo, and using all these different machines. If you already do CD34s, this will be fairly similar to you. At the end of this stage, after you've done your training, you want your log depletion to be close to 4. I don't think it will be at that time, but it should be close. Your documentation is nearing your approval, so your operations and QC documents, your batch record, your assays. You want to define the FIO data that you want to capture. We have to report a whole data sheet of additional cell types to the clinical team, so it's important to decide what you want to capture because when we're sending off this product, this is the last chance you're going to get before it goes into the person. And also I would develop a cryopreservation procedure. Again, if your facility doesn't have one already, this would be the time to do the development. After that's completed and you're satisfied, you move on to the engineering stage. This must be at least 3 runs. You're going to start off with an engineering protocol with clear perspective goals, and usually those goals are going to be somewhat aligned with what your IND, IDE state for your final product. I would also write a stability study for the fresh final product. So define how long you want that fresh final product to sit when it's formulated. Like I said, we're infusing from formulation at 8 a.m. to infusion 2 to 3 in the afternoon, so only a couple hours. But you have to ask yourself, do you want to go out past 24 hours? I think it might be important. We haven't had this happen, but a patient maybe runs a fever or something, you might have to go to the next day. So it doesn't hurt to have a longer expiration. At the end of this stage, you want to see the log depletion improve. You want all of your documentation, operations, and QC approved. And you want an expiration defined for your fresh final product. And I'm going to highlight again the QC assay. Extremely important at this stage that you have completed your development of the assay. This is how you're going to gauge success. And when you're looking at this assay, the TCR alpha beta cells are going to be only a handful of events per 100,000 cells. So it's really important that you know that your assay is capturing all of those events and you're seeing everything, because this is what we're trying to do. And again, if Miltenyi was a good resource to us as well for developing our assay, as long as LCGM, of course. After that stage, we'll move on to process qualification. Again, 3 runs. The qualification protocol must exactly match your IND specifications for your final product. At this time, I would also recommend doing a stability study for your frozen final product. So I would do 2 at the same time in the sense of a long-term stability, 1 to 2 years, and an in-use, meaning post-thaw. So when I say post-thaw, I mean you want a non-diluted, so go to the bedside, thaw the product, and infuse the patient, which is usually going to be 30 minutes to an hour, and then a diluted expiration, meaning you've diluted it with Normasol HSA or whatever you use in your facility, and you can extend out the expiration of that final product. The reason I say that is in case you have a bag break or something on the patient unit, you need a way to salvage the final product and also extend that expiration so the cells don't start dying. So at the end of this stage, you want your log depletion greater than 4, and CD34 recovery, which I hadn't mentioned earlier, greater than 60%. That's usually the low threshold that we're setting for this. All of your documentation is finalized, and your long-term frozen stability testing has started. You're not going to have results, obviously, because you're waiting 1 to 2 years. If you were to need to treat a patient earlier, you could most likely do an interim stability study or interim stability report and file that with the FDA. So after you completed all that, you'll move on to clinical readiness. This is a stage that we do where we're essentially going through a checkbox and we're checking off everything is completed. All the reports are finalized. All the stakeholders have signed everything that they need to sign. We're making sure that the documentation is done. I would do a dry run, picking up products from apheresis and bringing them back to your facility, checking that your chain of custody is kept from your apheresis facility to yourself, then back to the patient unit. Again, that's assuming if you're a new facility. If you're already a cell lab, you probably have procedures for this already, but it's just important to make sure everything works and that all the documentation works. And then, along with documentation, make sure that's all been filed with the agency and you're ready to move forward. At the end of this stage, your tech transfer is completed. I didn't mention it as well, but you would have QA documentation moving through at the same time, batch record issuance, label forms, all things like that. And if you use ISBT labels, I actually included our ISBT label codes just to make it easier because they can be hard to find sometimes. So this is our data from our tech transfer in terms of we did a tech transfer originally where we did a comparability with LCGM, and we were using a slightly different LOVO procedure. Our first 4 patient runs, our log depletion was 3.79 and our recovery was 62%. We determined that that wasn't good enough, and we wanted to align with LCGM. So we changed the LOVO programming that we had originally started with, and I'll get into that in the next slide, what I mean by all-in-one and separated. But for the next 6 patient runs, we did a qualification as well, but for the 6 patient runs post-qualification, our log depletion went up to 3.98, and our recovery went up almost 10%. So we saw a pretty big improvement. Usually, like I said, 4 is the benchmark that you want for your log depletion. You're usually going to make a patient dose around that, depending upon the patient's weight. So that was early to late 2022. So now, second half of 2023, our last 6 patients. So this is the team now getting comfortable with the process. They're becoming more proficient. They're becoming faster. So we haven't changed anything, but our last 6 patient runs, our log depletion went down to 4.57. Our recovery is about the same as 75%. I did this to highlight because when we were doing the requalification and kind of having issues at the start, one thing that Eleni had said to us is that this is more of an art than a science, and it was hard to understand that, considering that this is so automated. But as our training went up and we did more and more processes, our log depletions got better and better. So I just want to highlight that when your facility is getting started, it's going to take time to get to these high numbers. So don't expect it out of the gate. And I want to thank Eleni for sticking with us on that. So final observations. One thing that we've seen, we're running a split process and we're running in parallel. So everything should be the same in terms of log depletion and recovery. But we find that that doesn't happen. I'd say maybe 1 out of every 5 to 10 runs, we'll see a fairly large variance in the log depletion between the two samples. We investigated, we looked at personnel, machines, materials, everything, and there was nothing that we could find. It may be due to patient variability, but we weren't able to come up with an answer. So I just want to highlight that if your facility does run into this issue and you do an investigation, you say, why can't we find the issue? Well, we weren't able to find it either, so it's just something that could happen. The LOVO all-in-one procedure. So earlier when I was describing the LOVO, what happens is you load the cells on, the cells wash, and then they're harvested into a transfer pack, and that's it. At the beginning, we were using what's called an all-in-one procedure. They call it max shake, I believe is what they call it. So the cells never leave the LOVO during that entire cell washing procedure. So instead of going to the transfer pack, they'll stay in the in-process bag, which would be the number 2 actually right there, right here. So they'll stay in that bag, and you'll use their pouch and filter kit, and you'll inject the vials directly into the LOVO. You hemostat the bag, and you let it rock on the LOVO. It counts the time for you, and when it's done, it automatically washes the vial out, washes the excess reagent out. So we're not sure if that was one of the contributing issues to why our log depletion was low. It might be due to just like the bag is a different size, maybe due to just the washing procedures that we were using, because some of the specifications were slightly different. We weren't able to tell. So we just decided to match LCGM. It made more sense. LCGM has had a long track record of successful depletion, so, and also for our regulatory filings. It's just easier if we're doing the same thing. I've used that procedure on the LOVO for selections, and I've gotten very good recoveries, 60, 70, 80%. That was for like CD3, CD4, CD8 selections. So it works, but it just wasn't ideal for what we were doing at the time. Some of our future plans, I'd like to replace that final centrifugation with a LOVO wash. It would save us time, and it takes away the risk of bag breakage. We have 500 milliliters in a 600 ml transfer pack, and you're centrifuging that. There's high risk that that bag could break. It hasn't happened, but knock on wood, but it could. So I'd prefer to use the LOVO wash. It's automated and faster and safer. And like I said earlier, I'd like to extend our fresh product stability. I'd just like to take out the the risk in case something were to happen, and let's say pushing a patient infusion to like 8 p.m. or something like that, just in case. So something for the future. I'd like to thank my team. This is from our Christmas celebration. So our operations and scientific director, Catherine McIntyre. My team, which processes all the cells, Jemima, Guinevere, Malai, and Henry. Our QA lead, Tran. The QA leadership, Mary and Tamari. The QC team, supervisor, Zoe Amiri, and Jin, Hasib, and Helen. And then our medical director, Robert Losky. So that's it. Thank you, everybody.
Thank you very much for inviting me in this wonderful event. I really apologize that I could not be there in person. I just returned from Sweden, though, 4 hours ago, so it was a very tight schedule on my end. So I will talk to you about the evolution of the hematopoietic stem cell transplantation, let's say, and the graft engineering approaches that we have generated in the Clinimax systems. And then I will focus more on the TCR alpha beta depletion process and on the Clinimax Prodigy mostly, and continue with the flow cytometric analysis and the closing remarks. So the isolation, the depletion, or the enrichment of immune cells and hematopoietic stem cells goes many years back, actually 1997, where we had the first medical device approval in European Union for our Clinimax CD34 system. Eventually, we have a respective, let's say, approval from also in the US. And for the CD34 enrichment, we're basically enriching for the hematopoietic stem cells, and our transplant consists only of the hematopoietic stem cells. Originally, we decided to transfer the process from the Plus on the Prodigy. And what you can see here is basically a comparison that was done between the process on the Plus and the process on the Prodigy from one of our external collaborators, which was also published. And what you see here in pink is actually the hands-off. So you see that the Prodigy has the ability to, or offers, if you like, the possibility for the lab to have less operator interaction with the machine. And as a general remark, we started with the Clinimax Plus, then we moved to the Clinimax Prodigy, and we first applied or tried to adapt the processes of the Plus on the Clinimax Prodigy on the tubing set 310. And eventually, we transferred slowly the process on the tubing set 320. The 320 versus the 310 have several differences. This is not the correct forum now to discuss this. But let's put it like this. One of the largest differences is the size of the chamber between the two tubing sets. And also as a general remark, the processes that we are doing on the Clinimax Prodigy on the tubing set 320 are more equivalent to the Clinimax Plus compared to the processes we have released on tubing set 310. Of course, the CD34 enrichment was a very successful therapeutic strategy to mitigate GvHD. However, the absence of T cells was associated with more rejection and relapse. We also lost the GVL effect and so on. So then we decided, or there was the idea to deplete the CD3 and CD19. So then our transplant started containing not only the hematopoietic stem cells, but a lot of other lineages of the immune cells. And of course, inspired by Tübingen, which I believe some researchers and pediatricians are already sitting in the audience, a further advancement of the CD34 depletion was generated. And in this case, we went on to deplete for TCR alpha beta alloreactive T cells. And therefore, now the best hematopoietic stem cell graft is generated by depletion of TCR alpha beta cells and plus minus CD19 cells. So to make a long story short, we have a long-standing history in developing processes to generate improved grafts for hematopoietic stem cell transplantation. And I will now start focusing on the TCR alpha beta CD19 depletion system, which I would like to remind you that it follows this path. And also, additionally, because you will see it later on, we have the CD45RA depletion system, which we initially developed on tubing set 510. So moving on for the TCR alpha beta, I will present to you the first data that we generated when we slowly transferred the process from the Plus to the Prodigy on tubing set 310. But when we say Clinimax Prodigy TCR alpha beta CD19 depletion system means a existence of several different components. For example, we have the reagent, we have the tubing sets, we have the buffer, we have the Prodigy, and also we have the software on the Prodigy, so the application software. And back then, actually this process was generated before I was joining Miltenyi. We had a TCR alpha beta depletion manual panel to characterize the graft that was being generated by the Clinimax Prodigy. And when we started to disseminate the process to our external collaborators, these are the results we got. As goes without saying that we have a large and a normal scale, depending on the number of the cells that need to be labeled. We had a log TCR alpha beta depletion for the large scale of 4.26. For the normal scale, it was close to 4, so 3.99. And our external collaborators produced these results 4.42 and 4.22. And indeed, there was also a paper that was published in 2019 in which it says that the TC-alpha-beta depletion on the Prodigy on tubing Z310 was an average of 4.21. Moving on now to the tubing Z320, I would like first to clarify that on tubing Z320, we are now generating a next generation Clinimax Prodigy graft engineering approaches. And this is now a combined depletion where we combine TC-alpha-beta, CD19, and CD45 array depletion. Now, this is a more complicated system. Of course, it has a series of different reagents, TC-alpha-beta biotin and antibiotin, the CD19 reagent, the CD45 reagent, the tubing set with a larger chamber, the buffer, the Prodigy with the application software, and furthermore, we have developed and let's say made the flow cytometric analysis more robust, more automated, and easier for the end users by having at least two antibody panels, the immune cell composition and the frequency panel, which we use to characterize the grafts that are generated in these cases. This combined process is as combined as well as versatile. So you can choose whether you want to deplete only CD45 array or only TC-alpha-beta or TC-alpha-beta plus 19 or CD45 array plus TC-alpha-beta or CD45 array plus TC-alpha-beta and CD19. So it is a completely flexible process and of course depends on what the physician would like to administer to the patient. And the specifications of the process are shown here. This process is currently being developed actually, and we're planning at least to release it in Europe in the end of 2024. However, we have already disseminated this process in the context of a beta test to our collaborators in Frankfurt, and they returned to us the results that was in average a 4-log depletion of TC-alpha-beta cells, which as far as I heard Nick saying before me, this is what you basically want to achieve. So to summarize, this is a paper which was published, I think in 2018, which makes a large summary of a lot of hundreds, if I recall, even thousands of processes of depletions on the Clinimax Plus. And the average of CD3 depletion was 4.01 with a standard deviation of 0.4. We have at least one paper for the tubing for the Prodigy, the TC-alpha-beta on the Prodigy on tubing Z310 with this result. Mean depletion log TC-alpha-beta depletion 4.2 and standard deviation of 0.19. And we have at least one paper which was very recently published for the new process, the combined process on tubing Z320, with a mean depletion, log depletion of TC-alpha-beta of 4 and the standard deviation of 0.4. So now, why do our collaborators agree with what we are generating in-house? And the answer to this is because we have, during the years, developed and adapted the flow cytometric analysis to fit the difficulties that someone faces when they have to analyze TC-alpha-beta depleted grafts. So basically, there are 4 pillars that we have focused our flow cytometric analysis. Classical things, let's say, device setup, staining panel, the event acquisition, and the data analysis. So if we now have a look on the device setup, I will not say this is not rocket science, actually. Of course, you need the calibration of the cytometer, you need the compensation, you need the threshold setting in a good way, and you need to adjust the flow rate. But I would like to focus here a little bit on the calibration, and I would like to make this figure here a little bigger, and let's remind to ourselves a little bit of statistics and mathematics. Basically, what it shows you here is that 2 different populations which have been stained with a fluorochrome, and what you want to achieve is a maximum difference between your stained and your unstained sample. And also, you want to have samples which show low CV, so low coefficients of variation, because in all these curves, as you see here, these curves have all the same average, all the same mean, but the distribution is different. So you want to have as much as possible low CV in your flow cytometric analysis. For this, an optimal staining panel needs to be established. So how do we establish this optimal panel? First, we classify the conjugates based on their brightness. We use the brightest conjugates for the rare cells and for the markers of choice, the markers of interest, if you like. We used moderate conjugates for exclusion markers, not depletion markers, exclusion markers. We use the most Dinh conjugate for known lineage markers, such as CD45. We, of course, did the dead cell marker and the exclusion of dead cells, and we were very certain that we have a reduction of the unspecific background, which is currently done with the FCR blocking, but maybe you don't know, but Eleni has a series of antibodies, which we call rare antibodies, which do not need FCR blocking. So, for example, this is an analysis with a fluorochrome, which is how it should be, because the difference between your negative and your positive population is enough to set a nice gate, and this is how it should not be. And another example, and I would like to mention here, because this is something that most, sometimes it kind of is missed and it's not understood or seen. This is the same sample which was stained with the same antibody, which had the same conjugate, but it had a different conjugation method. So even if you have the same clone of the same, that you have an antibody with the same clone, even if you use the same conjugate, keep in mind that you have to collaborate with researchers who can really guarantee that the results you are seeing are indeed the results you are seeing. So do not feel comfortable that because you have, for example, a CD3 antibody, which is the same clone through different providers, it will perform in the same way, even when the conjugate is the same. This is my point here. Another example I would like to bring into your attention, this is the FMOs, these are fluorescent -1. Examples, for example, you have here the pre-depletion, and you have your T alpha beta cells and your T gamma delta cells. And of course, post-depletion, the T alpha beta population is no longer there. However, when you do your FMO, and you see that your FMO has, for example, 11 events versus your actual sample, which has 15 events, this is a problem. Your FMO actually shows that there is no difference, actual difference between the FMO and this actually stained sample. So this is not a good antibody and it should be changed. So when we moved on to the antibody panels, we spent a lot of time to optimize not only the panels, but the clones and the conjugates. And before I show you one example of how we do the analysis, let's discuss a little bit what makes this type of flow analysis so delicate. And the answer is actually that when the chemical composition of this alpha beta CD19 depleted cell product has 1% of CD34 stem cells, but because of the depletion has a 0.001% of T alpha beta T cells and a 0.008% of B cells. So basically you have only one T cell and T alpha beta cell in 100,000. Therefore, how many events would need to be acquired for a safe result? So basically, we are now in the area of rare cell analysis. I will not go a lot into mathematics. We can have this discussion anyway offline anytime. So basically, the rare cell analysis follows a Poisson distribution, which you see here, that it's a shifted distribution. It's not a normal distribution. And the CV, the coefficients of variation, in the Poisson distribution is magnified. I will not go into the details, but we can always discuss this. As a general remark, lower CV means higher reproducibility. So if you have 1 out of 100,000 cells, which was the case of the T alpha beta cells, and if you want to have a CV of 10%, if you want to acquire, let's say, approximately 100 events, you would have to measure 10 million cells. And this is not what I say, this is what mathematics say. Obviously, to measure 10 million cells would require a lot of time, which is not, let's say, applicable in the clinical setting. So therefore, measuring 1 million cells is a fair compromise. Measuring less than 1 million cells is actually risky and actually means or has the risk that the assay is not reproducible. So going now to apply this, or let's say knowledge or science on how we developed our assays for measuring and analyzing the T alpha beta depleted grafts. As I mentioned before, we have 2 panels, the IMCC panel, which is the immune cell composition panel, which you see here pre-depletion, and this is post-depletion. I will not go into the details of this panel in the interest of time. The obtained parameters we get from the IMCC is the cell concentration, the viability of all leukocytes, the graft composition, and the frequency of CD20 positive cells. So I will focus a little bit on the analysis of the frequency panel, which is actually the interest to measure the T alpha beta so we can then characterize the log depletion. We start first by selecting only low SSC cells. Then, of course, we go with a dump channel, which you see here. It's the 7AAD channel for the dead cell exclusion that also has the CD14 and CD15 markers here. So basically, in this case, we exclude, so let's say we take only the viable lymphocytes because we exclude all positive populations, which are not only the dead cells, but the CD14 and the CD15 cells in this case. And then, of course, we stain, then we further see the CD3 positive population here. We take the CD3 positive population, and then we backgate it again back to 7AD to have the official, let's say, viability of the CD3 cells. And then we split this plot here for the TCR alpha beta, TCR gamma delta, and CD45 array, CD45 RO, and eventually the CD45 RO are coming also by analyzing the percentage of the CD45 ROs also in the TCR alpha beta fraction. So this is how it looked pre-depletion, and this is how it looked post-depletion. And of course, the obtained parameters from panel F are the frequency of TCR alpha beta, TCR gamma delta, TCR alpha beta, CD45 array and RO, respectively, and so on, and the frequency and viability of CD34 cells. I have not gone into the details for that in the interest of time. So, to summarize, factors affecting flow cytometric analysis. You need a good device setup. You need the right choice of the antibody clone. You need the right choice of the conjugate. The brightness is a factor here. You need a very good gating strategy, exclusion of debris, exclusion of non-relevant lineages, reduction of unspecific background, use of appropriate samples, spiked samples to set the gates, backgating, good FMOs as additional controls, and it is also crucial to acquire enough events. So as a closing remark, T alpha beta depletion, at least from a Miltenyi perspective, has demonstrated a superb efficiency at the clinical setting. And when the use of proper medical devices and standardized flow cytometric analysis is achieved, for example, via express modes, we can further advance administration of T alpha beta depleted grafts for a plethora of diseases. By express mode, I mean a fixed template which someone can get to analyze directly the T alpha beta depleted grafts, at least on the markspan 16, or the markspan 10, which are the flow cytometers of the Miltenyi. And with this, I thank you for your attention.
Hi, everybody. My name is James Dinh, and I'm here to introduce you all to the TCR alpha beta CD19 depletions that we have done on the Clinimax Prodigy here at Stanford's Laboratory for Cell and Gene Medicine, otherwise known as LCGM. So just to introduce LCGM, as a facility, we're a state-of-the-art cGMP facility supporting clinical investigators during early phase preclinical development through clinical manufacturing for first-in-human therapies. Our mission is to develop innovative manufacturing platforms for the GMP manufacturing of cell and gene therapy products, enabling Stanford investigators to bridge the bench-to-bedside gap for treating patients with large unmet medical needs. Our facility contains a clean room space of about 3,300 square feet with a total of 6 clean rooms. We have rooms for cryopreservation, LN2 freezers for storage, an X-ray irradiator, and space for PD work. So here's just a quick outline of what I'll be going over in this presentation, starting with the T-cell alpha beta depletion process flowchart on the Prodigy platform. I'll be diving into some of the major differences between our clinical process and the Prodigy process, and what those final formulation steps may look like after sealing off the target cell fraction bag. I'll begin to wrap up our presentation on the goals we've set when transitioning our current process onto the Prodigy, including the results we received at LCGM as in addition to past published data. And lastly, to conclude this presentation with our ideas of process improvements moving forward. So in comparison to our current depletion process, the Clinimax Prodigy has simplified the entire process onto a single platform. Once we receive our apheresis, operators determine whether to proceed with a normal scale or a large scale process based on the starting TNC and the apheresis. After making that decision, the Miltenyi TS310 tubing set is installed onto the Prodigy alongside the proper buffers, reagents, apheresis, and then the Prodigy will pretty much take care of the rest. Operators are responsible for inputting specific cellular parameters, such as the starting WBC concentration, our starting volume, and the frequency of total labeled cells, but after that, the process is hands-off for the rest of the depletion. The next morning, operators will return to the Prodigy with the target cell fraction bag ready to be sealed off, centrifuge down, and ready for final formulation steps, whether it's to be prepped for cryopreservation or prepped for fresh infusion. And here's a short table. I'll quickly be going over some of the major differences between our current process and the Clinimax Prodigy process, starting with the equipment and tubing sets required for each process. Our current process requires both the Lovo cell processing system, which takes care of 1 platelet wash and 2 antibody washes. Therefore, the washes on the Lovo require 3 separate tubing sets. And then continuing onto the Clinimax Plus, this requires its own tubing set for the depletion portion of the process. Versus the Clinimax Prodigy, it takes care of the platelet wash, antibody washes, and the depletion process all within a single tubing set. Again, that's the Miltenyi's TS310 tubing set. As for labeling reagents that are utilized excuse me, within this depletion process, the current protocol requires operators to manually add each reagent vial via syringe and needle between each wash on the Lovo within a biosafety cabinet. When using the Prodigy, these glass vials can actually be directly spiked onto the tubing set upon installation, and then the proper valves will automatically open to add these reagents on the predetermined steps. As far as our process duration, the times listed are reflecting from the start of the platelet wash to the end of the depletion process. So with starting on the platelet wash on the Lovo and ending depletions on the Clinimax Plus, our depletion process takes about 4.5 hours, where the Prodigy estimates the time for the depletion process, whether it's a normal scale or a large scale process, to take anywhere between 9.5 hours to 10 hours based on our experience at LCGM. So one thing I'd like to note is that for the Prodigy, in addition to the 9.5 to 10 hours dedicated to the depletion, there's also the option to have an overnight hold before the harvest so that the cells can remain at 6 degrees Celsius overnight and allows the Prodigy to be left attended until the end process set time has been reached. This end process set time is set by our operators prior to starting the depletion for the next morning. As far as manual hands-on time, the current process requires about 8 hours from the operator, whereas the Prodigy process requires only about 3 hours of hands-on time. So although the Prodigy process might take a little bit longer than our current process, it requires a lot less manual operating. And that brings me to our last row, speaking of automation. With less automation on the Lovo Clinimax Plus method, there's a bit more open processing, which leads to more operator variability. On the Prodigy, there's a significant amount of more automation with more closed processing, and this can lead to less operator variability. This should also allow us to produce more consistent products outside of donor variability, but this means that there's less ability to troubleshoot or alter the process without investing in a customized application. In our current process right now, we load up to about 55 billion total nucleated cells per column or depletion to compensate for the limits of the Clinimax Plus. The pre-programmed TCR alpha beta depletion protocol that comes with the Prodigy has the option to run either a normal scale, which limits our starting TNC to 40 billion cells, or a large scale run that limits starting TNC to about 80 billion cells. And so what I like to emphasize on the upper half of this table is that the limit of the CD20 cells decreases for a normal scale process, which is lower than our current protocol. And this will limit us even more in regards to how many cells we can load per depletion. Just revisiting 20 of our last clinical runs using our current process, more than half of them have had a total of CD20 cells that exceed that 5 billion cell limit. So it'll cause us to think of how we'd like to move forward with processing, whether we would like to do 2 smaller normal scale runs or 2 large scale runs, or maybe a combination of a normal scale and a large scale run. But in the end, it really depends on how many cells we receive from the donor's collection. For the second half of the table, in our current clinical process, we typically use one vial of TCR alpha beta biotin reagent, 2 vials of antibiotin, and 2 vials of CD19 reagent. The numbers for the Prodigy process are actually hard-coded into this protocol based on whether you run a normal scale or a large scale run. So the normal scale requires only 1 vial of each reagent versus the large scale, which requires 2 vials of each reagent. And just to emphasize once again, once all of the required buffer bags, cleaning solutions, reagent vials, and the source bag is installed onto the tubing set, the depletion is a hands-off process once the operators start the separation. This will bring me to our final formulation steps. Pretty much post-depletion, whether it be from the Clinimax Plus or the Clinimax Prodigy, the final formulation steps are identical for both processes. Both processes produce a target cell fraction bag at the end of the depletion that gets sealed off of the tubing set, and then this target cell bag will be centrifuged down to a pellet. The supernatant will be removed via plasma extractor, and then we'll use 2% HSA plasmalite A or normal salt buffer to formulate the final product as we remove the final QC safety samples. On day 2, we have 3 options. It's either we prepare the final product for fresh infusion, we can infuse half of the final product fresh and then cryopreserve the second half, or we can cryopreserve the entire final product. So before setting any expectations for the TCR alpha beta depletion process on the Clinimax Prodigy, we revisited about 20 of our recent clinical processes at LCGM. Our TCR alpha beta log depletion ranges from about 3.60 to about 5.33 with a mean of 4.56. As for CD34 recovery, we have a broader range from about 57 to 93% with an average of being around 69%. And so we looked at previously published data from one of the first groups to perform a TCR alpha beta depletion on the Prodigy from the half strip group, and they came up with a mean of TCR alpha beta depletion, log depletion of 4.2 and a mean percent CD34 cell recovery of about 84%. So alongside of wanting a good NK cell and TCR gamma delta cell recovery to encourage GVL, our current process aims to get a CD34 dose of at least 10 million cells per kg, which is why we set our goal for the CD34% cell recovery to be equal to or greater than 60%. We also set our goal for TCR alpha beta log depletion to be greater than equal to 4.0 as to receive a TCR alpha beta dose of being less than or equal to 1 e to the 5th cells per kilogram, as these doses shall help aid the prevention of GVHD. So at LCGM, we were able to complete 3 normal scale processes and 3 large scale processes on the Prodigy. As you can see, the CD34% cell recovery has met our expected outcome of being greater than or equal to 60%. However, we've been experiencing a low TCR alpha beta log depletion in half of the runs one from our normal scale processes, and then 2 from our large scale processes. And then yes, this could be due to donor variability, but with half of our runs not meeting the expected outcome for TCR alpha beta log depletion, it meant that we would have to adjust our final product doses, meaning our hypothetical patient might not be able to receive as many CD34 cells as he or she should. However, by taking the average of all 6 Prodigy runs, the mean average is for CD34 cell recovery of 76%, and TCR alpha beta log depletion of 4.02 do reach our expected outcomes. In our patient runs, the cell numbers we load per column are usually closer to the maximum limit, which is why LCGM typically splits our initial apheresis collection, and then we run 2 separate columns per patient. So to address the specifications on the right, the Clinimax Prodigy process, the biotin reagent specifies that 1 vial should be able to cover a maximum of 24 billion TCR alpha beta cells and a maximum of 40 billion TNC. And looking back at our data, we loaded well below the maximum limit. As for the large scale process, the limits are listed as 45 billion for the total label cells, which includes the TCR alpha beta and CD19 cells. However, there's a maximum for the CD19 cells that caps at about 10 billion cells. So at the very least, the low limit threshold for the TCR alpha beta cells to be loaded would be around 35 billion. Again, we are underloading the amount of TCR alpha betas and are nowhere near this limit. So we revisited the paper by the Hashtrup group because the majority of their processes had a TCR alpha beta log depletion that was well over 4.0 besides 1. This is when we realized that they never reached the maximum limit for TCR alpha beta cells to load either. So it kind of brought us to this hypothesis or conclusion that maybe underloading the TCR alpha beta cells may encourage a better TCR alpha beta log depletion. Out of the 3 depletions that they performed using the normal scale process, the range for the TNC loaded was about between 29 to 38 billion TNC with the maximum of 40 billion. This consisted of a range of about 6.9 to 11 billion TCR alpha beta cells being loaded when the maximum limit was actually 24. So just to emphasize that is more than half of the maximum limit. Out of the 7 depletions that utilized the large scale process, the range for TNC loaded was from around 52 to 79 billion TNC with the maximum limit of 80 billion. And this consisted of a range of 9.2 to 24 billion TCR alpha beta cells, while the lower limit would be around 35 billion TCR alpha beta cells, also not really close to the limit. So although the Hashtrup group did have a higher starting TNC than we did, they somehow received a better TCR alpha beta log depletion than LCGM. And after revisiting this data, we found that they were also loading a significantly lower amount of TCR alpha beta cells when performing these depletions. However, certain results do kind of stick out to us, such as in the large scale runs where multiple depletions received a TCR alpha beta log depletion of about 4.2, even though they loaded different amounts of TCR alpha beta cells. Or the last run that is listed that received the lowest log depletion of about 3.98, but at the same time, they loaded one of the lowest amounts of TCR alpha beta cells at around 11.6 billion. But I would like to mention that the donor variability is a key factor, and that may be coming into play as well, but we are still investigating on how we can improve our TCR alpha beta log depletions here at LCGM. So thus far, we've drawn the conclusion that transitioning to the TCR alpha beta depletion process onto the Clinimax Prodigy has its benefits with increasing automation and reducing operator hands-on time and variability, which ultimately will lead to less training. However, the purpose of these TCR alpha beta depletions is to encourage and support a better engraftment and at the same time decrease the risk of GVHD. So LCGM is currently working alongside Miltenyi Biotech to figure out how we can improve our TCR alpha beta log depletions and improve our process as a whole. As far as process improvements, there may be a transition for the TCR alpha beta depletion process to use a newer MBI tubing set that contains a larger centrical unit. This may help improve each wash performed by the Prodigy. We'd also like to explore the possibility of eluting our final product with a 2% formulation buffer directly off of the Prodigy. Hopefully this will allow us to save some time and remove the final formulation steps I discussed earlier today, as it'll keep the process more automated, closed, and reduced operator variability even more. And then lastly, we are trying to determine the appropriate limits for the TCR alpha beta cells loaded in both normal and large scale processes in order to achieve our goal of a TCR alpha beta log depletion equal to or greater than 4. These limits will allow us to make the CD34 dose of being equal to or greater than 10 million cells per kilogram, and then the TCR alpha beta dose that's less than or equal to 1 e to the 5th cells per kilogram. And lastly, I just wanted to give a big thank you out to everyone who's spent the time working on this transition here with us at LCGM, from Dr. Patena and the clinical team, the folks at Miltenyi Biotech, especially Navneet, Zane, and Mitch for coming on site to assist us in person, as well as everyone in LCGM that has played a part into getting us where we are now, from PDM directors and associate directors, Gavin and Carrie, to our operators and verifiers, Garija, Satomu, Timothy, and Dean, and to our QC department, Fariba, Shreyashi, Julie, and Trupti. Thank you for your time.
Okay, thank you very much for all speakers for the great talk with lots of the details. Now we are open for the panel Q&A. So let's start with a couple of online questions. I think this question is originally from Nick, but anyone can chime in. So I know we spent lots of the focus on the alpha beta depletion, the log depletions, but here is a question about CD19, CD20 depletion. So the question is, the team has a very good alpha beta depletion, but we can never get CD19, CD20 depletion as well as on the climax as you did, Nick. So what kind of they can do to improve the CD19 depletion in general?
Yeah, the numbers that I put were only for TC alpha beta log depletion. Sorry, yeah. Sorry. Yeah, I'd have to pull the numbers for the CD19, CD20 log depletion, but all the ones that I put in my presentation were just for TC alpha beta. Is that what they were asking?
Yeah, so they want to know how to improve the CD19, CD20 in general.
Yeah, we don't focus on that. We're only focused on the TC alpha beta. We capture that data and it's important, but it's not the main concern. I think one thing to note as well. So James was talking about it, but the limit for the CD19 vials is 5 billion cells per vial. I think that it's important to know, like, with these limits, these are the limits for the process to work okay. I think that limit might be a little bit lower in reality, maybe around 3 to 4 billion cells per vial. So it could be adding another vial. That could help, but and we've shown that actually that will help with the TC alpha beta depletion as well. It helps with both. So just something to note.
Okay, thanks, Nick. So that's another question about the Lovo instrument. So they want to ask whether the Lovo instrument is inside of the hood or it's a benchtop instrument.
It's benchtop. So it's entirely closed. We can open up the tubing set outside of the BSC, and we just set it up outside. All you have to do is weld on your starting product bag, and we use a different transfer pack for our final product, and that's it. And so we just do the tube welding outside the BSC.
Okay. And the team is doing a manual bag wash inside of the hood, and except the platelet was on the Cope 2991. I'm not sure what the device is. So they want to ask if the Lovo can save the time compared to the manual wash.
Manual wash, I'm assuming, meaning centrifugation?
Yeah, I guess so. It's
Yeah.
manual wash inside of the hood for platelet wash.
Yeah, it definitely will. So we use, I'm going to compare it to the Cobe because that's what we use for our CD34 selections. Unless you have a quote-unquote automated procedure for the Cobe, it's operator dependent. We do ours where we're doing a semi-manual procedure where the operators are looking into the bag and watching the cells as they move towards the center of the bag, and they have to stop that in time. So between setup and watching that, it takes more time. Whereas the Lovo is weld the bags on, press start, and throughout the washing process, it'll ask you to mix the bag a few times here and there. But the Lovo is way more automated, way easier, and especially compared to centrifugation. I mean, you're going to have open conical tubes and things like that. It's a way better piece of equipment for cell washing, I think.
Okay, so there's another online question. So basically, when you do the parallel climax depletions, you have two machines. Mm-hmm. So is there a technical reason that not combine the target bag at the end?
Yeah, that was requested. Essentially, it gives more. What can end up happening is if we do end up having, like I said, one of those target bags with a much better log depletion, we don't want to end up mixing the two, and then suddenly we're meeting in the middle for the TCR alpha beta dose. We're going to want to infuse the best product possible. So if we check the two target bags first, then we can decide how to mix the products later. I know it adds essentially an extra QC portion, so you're going to do 2 samples as opposed to 1, but I think it gives you more options with the final product for formulation.
Yeah, understand. That's definitely reasonable. Any questions from the room?
I'm not sure if I'm going to digress from the topic of the technicalities of doing the depletion, but it's more of a real-world question about at what point does an institution take the stance of switching from a semi-automated to a completely automated, and what makes it cost-effective, and can it be justified not just for a top-notch institute like Stanford, but a lower and middle-income country with a socialized medicine, but a centralized way of delivering it? Yeah.
That's a good question.
That's probably a question for Alicia.
It's about my pig rig.
I think I need to answer this question. For us, the trigger of moving from the Clinimax to the Prodigy is exactly the idea of moving to a manufacturing which is less operator dependent, and then can be transferred to exactly other institutions and other countries where having the highly specialized personnel and that experience that all of us have heard in different talks is necessary to have a certain level of depletion is not going to be needed. To the best of my knowledge, the Prodigy is not yet widely used for manufacturing alpha beta T cells. And while we are doing this work, we are learning that, again, using an instrument which is so automated, yes, it gives you the possibility of achieving a result which would require some experience to get, but on the other hand, is not giving you the ability of driving that car based on your own experience, because there is a limit to that car, which is great for who doesn't have experience, but it might be a limiting factor for who has a lot of experience. So it's a balance, which I think that overall, for really making the use of this approach more wide, it's beneficial. It requires still some adjustment. And in fact, since we have Eleni on the other side of the monitor, I think that really, and we have Rupert and other Miltenian alpha beta depletion experts. I think that really, I just would like to open up the discussion about what can we do to improve the manufacturing with the Prodigy. Not just tomorrow with the runs that we are performing, but in general, what's our vision between now and the next 5 years? How I can make this manufacturing really optimized with an automated system that can be much more used from many other centers.
So thank you very much for opening this discussion. As you already may know or have understood, I'm a great fan of the Prodigy. And based on our internal data that we generated in-house during the development, but based also on the data that our collaborators have also generated, I do not see the Prodigy having a less performance compared to the Plus. Also, I think I mentioned this before, the Prodigy, even with your data that very nicely were presented, has a lower standard deviation. And this is what we are aiming for the Prodigy. Now, it is understandable that when you are researchers, physicians, developers who have been working with the Plus for years, who love the Plus, who have this wonderful experience, and you can play a little bit. It is true that when you take the Prodigy, you kind of feel that you lose this control. But basically, this is the first step. The more you work with a Prodigy, the more you will improve. You will see this gradually. So it's just a matter of embracing the device and working with it as much as possible, to make a long story short. It's more of a psychological, I would say, hurdle that there is there for the Prodigy. It's that you think that it's automated and you can get, let's say, a log depletion of 5. I'm not saying this is not possible, but it would be at the end with the same frequency as it would be on the Plus. So if you do the same approaches on the Prodigy for 10 years and you run it 5 times per week, you will see the numbers being somewhat like the Plus, but with less standard deviation. And this is important, I think. And to address, unfortunately, I cannot see the room because somehow my computer is not always collaborating with Zoom. Someone asked whether the Prodigy can be applicable for other middle countries. Yes, it can. And that depends on the number of transplantations that you are performing per month. So it's at the end, if this alpha beta is such a promising approach, it should be something that you should consider buying a Prodigy at least. Specifically, as Alicia said before, that if you specifically don't have a lot of experience, it would be the perfect device for you.
Okay, thank you very much. So I think this concludes our manufacturing section. Based on the schedule, we seem to have 15 minutes break before the winner of the best clinical abstract. Thank you.
Thank you very much.